All right. Good afternoon, everyone, and welcome to American Floral Endowment's Grow Pro webinar series. I'm your moderator once again, Dylan Sedmak. I'm a member of AFE's Young Professionals Council and also a regional account manager with Pan American Seed. I received my bachelor's in horticulture from The Ohio State University, and before my time at Pan Am, worked as both a grower and researcher in the industry. Today's session is on success with tissue culture. On behalf of the endowment, I'm excited to be a part of AFE's Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE sponsors. The views and opinions expressed in these webinars are those of the speakers and do not necessarily reflect the views or positions of AFE. As an independent nonprofit, AFE cannot endorse any specific product or opinion. If you're interested in becoming a sponsor for one of our GrowPro webinars, you can find that information on AFE's website on endowment.org/GrowPro. Today's session was pre-recorded in English by Dr. Dayton Wild. After the presentation, Dr. Wild will join us for a Q&A. Feel free to submit your questions to the Q&A feature or chat at any time. We'll answer as many as we can before the end of the hour. Unanswered questions may be answered through a separate email exchange. This session is being recorded and will be shared to AFE's YouTube account. Through YouTube's accessibility features, you can access closed captionings in other languages. To get us started, I'd like to share a bit about today's expert presenter. Dr. Dayton Wild is a professor at the University of Georgia with an emphasis on molecular genetics of ornamental plants. His research interests are in the application of molecular genetics to improve traits of ornamental plants, and his lab is currently using DNA sequence information to screen plant populations for natural variation or induced variation in candidate genes that control ornamental plants. He is currently working on the use of CRISPR to develop powdery mildew resistance in Gerbera.
Well, thank you for the introduction. Welcome to this webinar on plant tissue culture. In this presentation, I'd like to cover the following topics. Why would you want to use plant tissue culture? How do you optimize it? Plant growth in culture? How do you get the plants from culture out to the real world? And then I would like to give you an example of tissue culture that we're doing in our lab. We're working with 3 species now, Gerbera daisy, dogwood, and rose. The Gerbera daisy is presenting a challenge, and so that's really what I'd like to talk about a little bit today. And then finally finish up with some new tissue culture technology. So plant tissue culture takes advantage of this remarkable property of plant cells to reprogram and regenerate into whole plants. One way of doing this is through organogenesis, in which a cell, say a leaf cell, is reprogrammed to form shoot meristems, which then grow into shoots. These shoots can then be propagated in the thousands, and then another reprogramming goes on so that some of these shoot cells become roots, leaving you with an intact plant. First of all, I guess just to say, not all cells are capable of doing this. Not all plant species are capable of doing this. But there are a number that are, a number of ornamental plants that can be propagated by organogenesis. These include African violet, poinsettia, ficus, rose, a number of important ornamentals. An alternative to organogenesis is a process called somatic embryogenesis, in which, again, leaves, say, cells from a leaf or some other part of the plant can be reprogrammed to form cells that give rise to somatic embryos. These somatic embryos can be reduced into the thousands, and then when necessary, they can be germinated, just like seeds, to again form whole plants. So there are a number of ornamentals that can be propagated by somatic embryogenesis. These include clematis, sansevieria, rose, begonia. Some of these plants uh, were mentioned before as being able to be regenerated through organogenesis. And sometimes some cultivars respond to somatic embryogenesis, some respond to organogenesis. The advantages of using tissue culture are that they allow the clonal multiplication of hybrids and unique genotypes. They allow the large-scale production of disease-free plants. They allow you to control the timing of plant production. And because you only need to take your plants out when you're out planting your tissue culture plants, uh, you don't need the greenhouse space for stock plants. On the other hand, plant tissue culture is not for every operation or every plant. There's the cost of the facility for aseptic production to be considered. Uh, Trained personnel are required. There is the potential in culture for what's called somoclonal variation. And then finally, not all ornamental species or cultivars are amenable to in vitro regeneration. Despite this, there are over 150 ornamental genera that are propagated by tissue culture in commercial labs worldwide. Tissue culture is also critical for ornamental biotechnology. It's used for cryopreservation of material. In this case, tissue culture material like this somatic embryogenic culture of dogwood can be frozen in liquid nitrogen, where it can stay practically indefinitely. When it's needed, you can then this can be pulled out of liquid nitrogen storage. And what this shows here are somatic embryos regenerating after coming out of deep coming out of the deep freeze. Tissue culture is also needed for genetic transformation because genes are introduced into single cells and they then need to be regenerated into whole plants. And similarly, for gene editing, single cells are edited, and then again, those are regenerated into whole plants. So whether you're using tissue culture for biotechnology or for propagation of an ornamental species, there are several components that are very important to this process. The first is simply establishing cultures that are clean, bringing plants in from the outside into culture. Secondly, the type of explant or the type of tissue that's used to do this can give different results. Once you get this material into culture, it's important to maintain sterility because we are providing the plants with nutrients and carbohydrates that are also very attractive to bacteria and fungi. Let's see, another thing that's important is the type of tissue culture system that's being used. And then within the medium that we provide the plants, the nutrients are important as well as plant growth regulators. And then finally, environmental conditions such as light and temperature are also critical. And I'd like to go through these one by one. For starting cultures, they can be initiated from a number of different explants. This includes a shoot tip, shoot flowers, either flower parts like petals or the entire flower sometimes. They can be started with leaves or the petioles from leaves. Lateral buds are used. Sometimes stems and roots are also used. The age of the explant can be important to initiating a culture so that young leaves can be more responsive in culture than old leaves. Plants don't live alone. And when you bring them in the culture, you can see that they host diverse microorganisms. In this picture, you can see maybe one, two, three, four, five, six explants that were put into culture that also carried along, in this case, bacteria here, you can see, or fungi, you can see there. Sometimes you get yeast. So you have to take some care to get rid of these before putting these into culture. This can be done by surface sterilizing the explants, uh, often by serial treatments of ethanol and bleach and sterile water. Um, the sometimes, or actually quite often, a surfactant is needed to help with this process, and antibiotics and fungicide are sometimes used. So the concentrations and exposure times of these different sterilants have to be determined experimentally. Uh, and you can imagine that that a leaf would be different to sterilize than stems of a woody plant. So you have to balance sterilization with tissue health. And finally, sometimes, despite your best efforts, there can be internal contamination or bacteria, say, growing within a plant. Uh, Sometimes you can get rid of this. Sometimes you can live with it, and sometimes you just have to start over. So once you get plants into culture, you have to maintain that sterility. And this is done by sterilizing the media that's used, the instruments that are used, and generally using an autoclave like this. To work with plants, you have to create a sterile workbench. And this is commonly done using a laminar flow hood in which filtered air is blown from the back to the front of the hood, allowing no bacteria, no fungi, no fungal spores to enter, in which it stays clean if the proper sterile techniques are used to avoid contamination. And then lastly, you'll need a clean room or someplace to grow the cultures. So there are a couple of different tissue culture systems. Uh, Most common or very commonly used is this semi-solid medium, is solidified with agar, that's kept in containers or in plates, containers like this reusable box or a plate or tubes, or sometimes there's a disposable containers that can be used for culturing plants. These containers need to exclude microorganisms, but also allow a gas exchange and hold humidity and keep these plants in a high humidity environment. To do this, these plates or boxes are often wrapped with something like parafilm or surgical tape, paper surgical tape. We find that the vented lids are also sometimes very useful for in preventing contamination and also allowing gas exchange. There are other tissue culture systems, such as liquid suspension, that are sometimes used, particularly for the propagation of embryogenic cultures. These cultures are aerated on an orbital shaker. There's and sometimes you get some sort of combination of both by using what's called a temporary immersion bioreactor. Uh, Shown here, this is a Rita system. In this case, cultures are grown here, and then intermittently the nutrient liquid is brought to the top and then allowed to cover the plants and then strains back down. So in certain situations, this system works very well. So whether you're providing the plants with liquid medium or with solidified medium, you need to give the plants everything that's required for them to grow. This includes macronutrients such as nitrogen, potassium, calcium, magnesium. Also, trace elements. There's a number that are shown here. You need to provide them with vitamins, with iron, and sucrose, because these plants won't be able to produce all the carbohydrates they need. So, what's shown here is Murashugi and Scood medium, which is one of the most widely used media. This particular medium has been used for over 40 years. It's based on the composition of tobacco. Obviously, not all plants are like tobacco, and so you need to find the most appropriate medium for your own species, and this is determined experimentally. So other commonly used media are woody plant medium and ganwort medium. So also within this medium, plant growth regulators are used. These are very important for controlling the growth and development of the plants. Auxins are generally used. Auxins promote meristem development, cell expansion, and root initiation. And then commonly used auxins are 2,4-D, IBA, IAA, and picloram. Auxins are often used in combination with cytokinins, which stimulate cell division and shoot proliferation. And these cytokinins that are used and are often used are BAP, 2IP, kinetin, and metatopolin. And as to which of these works best for you, this is something that has to be worked out empirically. So like I said, their auxins and cytokinins are often used together, and the ratios have different of auxin cytokinin have different effects. The high cytokinin to auxin ratio causes root initiation, while low cytokinin to auxin ratios result in shoot initiation. And sometimes there are other growth regulators that are used. For example, gibberellins are sometimes used to promote stem elongation and flowering. Light quantity and light quality are important to plant tissue culture. Most plants are grown in 25 to 100 micromole per meter squared per second. And also on a light cycle of 16 hours light and 8 hours of dark. Embryogenic cultures, on the other hand, are generally grown in the dark, as you would imagine for something like a seed. So light quality is important too. LEDs have been looked at in the last couple of years. LEDs are of interest because they require less energy and they emit less heat than white fluorescent lights. There are specific wavelengths, such as blue or red, or combinations of blue and red, that have been found to improve plant growth. And these, again, need to be worked out for the species that you're interested in. Other environmental conditions include the temperature, which for most plants is around 25 degrees centigrade, about 77 degrees Fahrenheit for most plants. You can store some plant cultures at 4 degrees, and the advantages of this is that they can go longer without being having to be subcultured, months at a time, and which reduces the amount of uh, space and work that needs to be done with these plants. So when eventually these plants are going to be ready to move from the tissue culture to the greenhouse, in which case the plants have to adapt to ambient light and ambient humidity. So in culture, the high humidity tends to give leaves that have thinner cuticles. Uh, on the outside of culture, the natural light has a higher intensity than what's in culture. And finally, plants that are grown on sucrose-containing medium now have to transition to photosynthesis for their carbohydrates. So uh, generally what's done is to wash the plantlets to remove the agar and the sucrose because this would be very attractive to bacteria and fungi. Uh, these plants are then transferred to a sterile potting mix and then grown either under mist or in closed containers. This mist is intermittent so that and plants are generally weaned off the mist. And in containers, they can be opened and gradually exposing the plants to ambient humidity. If you're going to import or export tissue culture plants, you would need a phytosanitary certificate. But for just interstate movement, plant permits are not needed unless these plants are endangered species. So now I'd like to talk a little bit about some tissue culture work that's being done in our lab. Uh, the American Floral Endowment has funded a research project in our lab to develop Gerbera daisies with powdery mildew resistance that develop using gene editing. So to do this project required developing a tissue culture system for specific Gerbera cultivars. So we had to figure out, how to get them in the culture, how to identify which is the most regenerable explant, how to best propagate these shoots in culture, and how to optimize the organogenesis. So we introduced 4 different Gerbera cultivars into culture. These first 2, we were able to use seeds to introduce these in the culture. But for the second 2, we're actually the 2 we were more interested in. These came as plugs that we grew in the greenhouse, and then you had to use greenhouse materials to establish cultures. So we use 3 different explants. Uh, the capitulum, or which is the flower head of an Asteraceae plant, the floral stalk, or also better known as peduncle, and leaves. So the explants from greenhouse grown Gerber plants were really a challenge to clean up. Even though they came as clean plugs and grew in a clean greenhouse, they were still, we were getting contamination with fungi and bacteria. So we looked at a number of different procedures for getting rid of these, uh, including an extensive water wash, use of ethanol briefly for 15 seconds. We looked at the bleach, using bleach for between 1 and 25% for 1 to 15 minutes. We looked at including fungicides such as Captan or Rocol, either from a 20-minute exposure to overnight. And then we also looked at antibiotics such as timentin, silver thiocyanate, or a commercial mixture called plant preservative mixture, PPM, which is a broad spectrum antibiotic for for 1 to 2 hours of exposure. And after a while, we came up with a disinfestation protocol that looks like this. Washing the plants, washing bacteria, washing fungi, anything else off the plants seemed to be important. So we had an extensive wash under tap water. We then incubate the explants in sterile water containing 1 mg per liter of ppm for an hour, and then cultured the explants on media on mammoth medium containing 1 mg per liter of ppm. And with these procedures, we got about a quarter of our explants into culture without any contamination. So we got response from the different explants that we use for cultures in leaves, capitulum, and peduncles. The capitulums gave us the best response, and so we use these in the future. This is a shoot explant of gerbera. And just to remind you, gerbera uh, uh, is a basal rosette, so it doesn't really have a true stem. It just except for the flower stalks later on. So then we had plants in culture. We then had to optimize the shoot propagation from this. So we looked at a couple of things. We looked at the subculture cycle, which is how often these explants, or I'm sorry, these cultures, shoot cultures, needed to be moved to fresh medium. We looked at light quality and compared fluorescent lights with LEDs, and we looked at light intensity. And in some of these treatments, we saw a condition called hyperhydricity, which is a growth abnormality. And to control this, we looked at gelling agent concentration. We tried silver thiocyanate in the medium and also looked at vented lids. And so finally, what worked best for us was a transfer to fresh medium every 2 weeks, a 16-hour light, 8-hour dark photo period at somewhere between 23 and 25 degrees, growing these plants in low fluorescent lights, somewhere between 10 and 25 micromole per meter squared per second, and then using a gel-right or an agar medium concentration that was higher than we used to use. Again, this was to control hyperhydricity, but we were using it at 3.5 mg per liter. And then finally, we found that vented lids also helped to control the hyperhydricity and improve growth. We also, before going any further, I just want to mention that we did also look at somatic embryogenesis. We found that we could get somatic embryos induced infrequently on the auxin IAA, but we could never establish embryogenic cultures. And so we decided to focus our efforts on organogenesis. So for this, we used leaves from gerbera shoot cultures, and we looked at several parameters to optimize organogenesis. We looked at the tissue culture system, both at the using both semi-solid medium and this temporary immersion bioreactor. We looked at different nutrients. We looked at those of MS. We also looked at a commercial formulation called gerbera multiplication medium. And with some additional medium additions, such as amino acids and vitamins. Then we looked at plant growth regulators, including IAA, IBA, 2,4-D, and then the cytokinins, BAP, TDZ, and metatopolin. Now, what made gerbera so hard for us to work with was that it was very, very slow in culture. It developed very slowly. It took a long time to induce the regenerable tissue, a long time for that to become shoot cultures. And these are experiments that were replicated. They were multi variable, and it just took a long time to do these experiments, and so that it was hard to make progress for one experiment to end to optimize it and move on to the next one. But eventually, we found what worked for us was using a semi-solid medium in a GA7 box, one of those reusable plastic boxes, using MS medium with glutamine and vitamins added to it. For the PGRs, 0.1 mg per liter of IBA and 1 mg per liter of metatoplin seemed to give us the best response. Using a photoperiod of 16 hours of light, 8 hours of dark at 25 degrees worked best, and also using low fluorescent lighting, they seem to work well. What you can see here was what we were getting. This is a shoot induction on the side of a leaf. You can see a new leaf emerging here. So that we were able to get organogenesis at a fairly good rate. We can now use this procedure to introduce genes and regenerate transgenic plants. So we use this system to regenerate plants after introducing two genes into plant cells. One gene is called green fluorescent protein, which allows the transformed cells to be visualized. And the second gene is hygromycin resistance gene, which allowed only the transformed cells to grow. I showed an example here. In this plate are leaves, pieces, and petioles that are growing in culture in the absence of hygromycin. In the presence of 10 micrograms per mil of hygromycin, these cultures die. So anything that grows from this is considered to be a transgenic plant or transgenic cell. So this is a leaf that's been exposed to agrobacterium with the green fluorescent protein, and this is you can see here these tiny constellations of green light show that these cells have been transformed, and then eventually some of these will start dividing and getting in the presence of hygromycin, so that you have hygromycin resistant cells that are grown here. And you can see that these also, this regenerating material is also showing green fluorescent protein expression. So that we had a transformation system. We then use this to edit Gerbera and work out a gene editing system for Gerbera. To do this, we use a model gene called PDS that gives an albino phenotype when it's inactivated. So in this case, we use a PDS gene from one of the cultivars we were working with to design a gene editing construct that was then introduced into Gerbera leaves using agrobacterium. And the gene editing of the Gerbera PDS gene, as you can see here, resulted in the production of albino shoots. So a similar approach is underway to edit Gerbera, the Gerbera MLO gene. Okay, so now I'll talk a little bit about some new tissue culture technology. And I mentioned at the beginning of this webinar that explants are able to be reprogrammed into cells that can give rise to shoots or become somatic embryos. So the question that's been asked for decades really is how does a leaf cell reprogram and develop into a shoot or an embryo. And in the last 10 years or so, I think we've gotten at least a partial answer to this. The genes have been discovered that control shoot initiation and somatic embryo initiation. These include genes such as baby boom, plethora, Wuschel, and growth regulating factor. So, with this information, we can now use these genes to try to enhance regeneration. And I'll show you an example here. In this case, these are explants from wheat, from grape, and from citrus that are on a medium that is used to induce organogenesis. This is when GRF is included, you get a much better, much more robust response of organogenesis for all three species. So this technology is promising for plant species or plant genotypes that don't regenerate very easily. In this case, though, the GRF was introduced into these different plants as transgenic plants. We won't be able to do that in the long term. So there are non-transgenic approaches that are for delivering these developmental regulators that are being explored. This both may be introducing these using nanoparticles or also a viral delivery. So in summary then, tissue culture can be used for the large-scale propagation of clonal, disease-free ornamental plants. Factors such as the culture system, the nutrient media, growth regulators, and light all need to be optimized experimentally for each plant species. And then finally, some plant species or within some cultivars within plant species are recalcitrant to regeneration in vitro, but new technology is being developed to address this. So I would like to acknowledge the American Floral Endowment for supporting our project in Gerbera, and also supporting Dr. Heather Gladfelder, who's a postdoc in my lab who did all the Gerbera work. And with that, I'd be happy to take any questions.
All right. Thank you, Dr. Wild, for your presentation. So before we jump into our Q&A, I'd like to just encourage everyone to join us next month for Dr. Charlie Hall and Dr. Melinda Knuth's presentation on trends to watch in 2025. And that'll be on Tuesday, November 19th at 1 p.m. Eastern. And the link to register will be in the chat. So now I'd like to open up the session for Q&A. Let's see here. So one of our first questions in the Q&A is, you mentioned that a low auxin to cytokinin ratio promotes shoot growth. However, I had the impression that a higher auxin to cytokinin ratio promotes shoot initiation. Could you clarify how the balance between these two hormones influences shoot formation?
Well, it's mainly driven by cytokinins. That and to promote shoot growth, you're going to need some auxin once you have shoot cultures established to maintain that growth. But it's really the higher cytokinin initially that's used to get the shoots initiated.
Thank you. And then another one here is soma clonal variation a problem for plant tissue culture?
Well, let's see. So simulclonation is caused by changes in genes or the regulation of those genes in ornamental plants, and this variation could be good or it could be bad. You may get a new trait like variegation from simulclonal variation, but if you want uniform plants, it's something that would be undesirable. But it doesn't happen at that high of a frequency, maybe around 1% or so. So not a big problem, and like I said, it can be can be advantageous. There are things you can do to reduce climaclonal variation, like reducing the time that plants are are in culture. Uh, because the longer there you have them in culture, the more likely you're going to get some accumulate these mutations. So that's why uh, mechanisms for storage like cryo or uh, cold storage are being looked at.
All right. And then any recommendations of acclimation temps for stage three TC items? And what should they shoot for temperature and humidity when acclimating?
Well, you're going to be looking for ambient conditions, which uh, would be, lowering the humidity to to what we have on the outside uh, uh, by gradually opening a tissue culture box or container, or by using mist and then reducing it slowly. Uh, the what the exact procedures you're going to use probably will depend on what kind of plants you're using too.
And then another question is, what should the temperature of the water be to clean tissue culture plants when removing agar prior to transplanting?
Probably whatever comes out of the tap. It's not, that's something that's probably not terribly critical. You could, use room temperature water.
All right. And what are the impacts of light level of a two sorry. What are the impacts of a high light level for TC growth? For example, 150 to 300 micromoles.
It depends on the plant species. I think that a lot of the plants I work with, something like that might lead to hyperhydric plants, vitrification in plants, which is obviously something you want to avoid.
Okay. And then are there any suggestions on how to maintain plants on cold storage? For example, the meat type of media or lower sucrose, lower nutrients.
Right. So they're not going to be growing as vigorously as they would be at room temperature. So you could get away with maybe half MS, or something of lower strength. Um, the but really what you're trying to do is keep the plants in a condition they're not going to be doing much growing at all. So they're and they're going to be large. Well, they could be in the dark too, um, but they're uh, mostly just this medium is not going to be terribly different. You're just not going to be using it as often as you would in a normal tissue culture situation.
All right. And then our next question is, does ppm used in Gerbera initiation eliminate contamination or just suppress contamination?
It eventually will we get them off of uh, 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 ppm, so they're it does not it does more than just suppress contamination. It will eliminate, at least in our hands. It's been very helpful for that.
All right. And another question pertaining to the transition stage for TC. So, should we be sterilizing our media, in our case, Elipot soil? And what's the best way to go about doing that? Presumably, when you're transplanting your stage three or what have you.
I think yeah, because yes, I would say it would be helpful. You have a lot of invested in plants up to that point. You want to make sure that there's not any problems with uh, things other than plants growing in your media.
All right.
Mhm.
And is there a standard time frame recommendation for being in TC that will minimize the risk of somoclonal variation?
No, I'm not sure there's a standard time. uh, it would, let's see. Um, I think that you do start losing regeneration capacity after a while, if you had cultures in a while. Uh, the Probably the shorter the better would be helpful for avoiding some clonal variation.
And then any success growing ornamental grasses in culture? And if so, what are the most detrimental steps during sterilization?
For grasses, to be honest, I'm not as familiar with grasses. Obviously, there's been a lot of work done with things like wheat that would be useful to follow, but I don't have any experience with grasses.
Here's a good one. If a single plant out of a segregating line was propagated by tissue culture, then the resulting plants were self-pollinated to produce seeds. Would the plants grown from these seeds be true to type?
If it's self-compatible, uh, yes, I would expect so, that there would be very little variation, yeah, that you'd find. But again, the plants, they would have to be able to cross themselves.
All right. And then again, back to the theme of sort of transplanting the TC, should there be a starter charge in the soil or media when acclimating to the greenhouse?
Probably, yeah, it could be helpful. Uh, it, each sort of tissue culture and greenhouse situation is somewhat different, but you could see how that might be useful.
Okay, and any specific considerations for the TC crops received with minimum or no roots?
For initiating these in the culture, or um, for taking these uh, after you've produced these.
I would presume that they're talking about receiving in TC plants in the greenhouse.
I suspect that if you were trying to get these to root, then so treatments with IBA or some other auxin would be helpful.
And we answered this question. Okay, the next one is, are antibiotics more effective compared to PPM?
It depends on maybe what you're trying to control. Uh, PPM is actually works against both fungi and bacteria, so that's helpful. Uh, there may be some bacteria that are more responsive to certain antibiotics like timentin. But again, in our hands, we found PPM to be very useful.
Okay, and another one is, what would be the best PGR for, I believe this is shoot initiation for Azalea Encore?
I would try cytokinins, like BAP or metatopolin could be helpful. Um, uh, I've never we do some work with azalea, but not in culture, hard for me to answer that. You just have to try.
And then is there any research or trials being done with shrubs from shrub production from TC?
I suspect so. That a lot of shrubs can be produced through rooted cuttings, and so that's they would not necessarily go through TC. Uh, but I would be surprised if there isn't something that like that has been done. But I'm not coming up with it right now.
All right. And then also, are there any commercial facilities to outsource tissue culture work?
There are a couple, mostly on the coast, in I think New York, Washington, California, Florida. There's a couple options that you could use to if you are interested in doing tissue culture, and but didn't want to have invest in the facilities yourself, you can certainly, I think there are places where you could outsource that.
And another here is what is hyperhidricity and how is it avoided?
Okay, well, hyperhidricity it's a physiological disorder uh, caused in tissue culture that leads to thickened stems and short internodes and sort of glassy or abnormal leaves. Uh, and it's usually caused by excess moisture in the medium or in the atmosphere. Uh, there's other, uh, it can also be caused up by a buildup of carbon dioxide or ethylene. More immediate components, sometimes like excessive cytokinins or nitrogen, can sometimes uh, cause hyperhidricity. So, in our case, we saw it, when we were doing some work in Gerbera with LEDs. We thought that would be great, but somehow it did lead to hyperhydric plants. Uh, that one of the reasons we went back to fluorescent uh, lights for that. But again, also just using the vented lids helped provide an atmosphere within the tissue culture container that was better for growth and avoided hyperhidricity.
All right. And can shoots be recovered once vitrified?
Yes, I think you can get out of, you can by adjusting the tissue culture conditions, you can get more normal shoots out of that.
I think that's all the questions we have right now. Just one last call out for any questions for Dr. Wild while we have him. Otherwise, we'll go to make some closing statements. All right. Well, first off, thank you very much, Dr. Wild, for your time and your presentation. Really appreciate it and learned quite a bit. And thank you to everyone for joining us today for another session of AFE's Grow Pro webinar series. And remember to join us next month for Dr. Charlie Paul and Dr. Belinda Knuth's presentation on trends to watch in 2025, which again takes place on Tuesday, the 19th of November at 1 p.m. Eastern, which you can register to participate in at endowment.org/GrowPro. While there, just make sure to go ahead and check out our past webinar recordings, other grower-related resources, and research reports available to you free thanks to industry support. We ask that you please complete the brief survey about today's session, where you can suggest additional topics and help us continue to improve these webinars. And thank you again for joining us, everyone, and have an excellent day.